Hi, everyone. This is Anqi Qiu from National University of Singapore. I'm delighted to introduce some of our work on nature and nurture, evidence from neuroimaging and genetics. As we are aware, the prevalence of mental illness tends to significantly rise from the age of 5 and onwards. However, our understanding of clinical diagnosis of mental illness in children below the age of 4 remains limited. This highlights the importance of exploring the developmental origin of mental illness. To date, our understanding of environmental factors and biological processes that influence brain development during early life, which may contribute to mental illness later in life, is still far from complete. Further research and exploration are necessary to bridge this knowledge gap. In 2009, my group and colleagues at the National University of Singapore initialized a birth cohort study called Growing Up in Singapore Towards Healthy Outcomes, GUSTO. GUSTO recruited women who visited 2 major hospitals, KK Women and Children Hospital and National University Hospital in Singapore during the first trimester of pregnancy between 2009 and 2010. Women had no history of mental health problems and metabolic problems prior to pregnancy. The sample is representative of Singapore population in terms of ethnicity distribution. The GUSTO study deep phenotype the environmental factors from pregnancy onwards. The environmental factors include complications during pregnancy, maternal mood and nutrition, family factors such as socioeconomic status, family support. The outcome measures in the GUSTO study are mainly on children, including metabolic measures, cognition, brain imaging, psychopathology, as well as epigenetics. At birth and infancy, we employed various techniques such as video recording, EEG, ERP, and MR to measure infant memory, executive function, and emotion functions, as well as brain structure and function. After 2 years of age, children are followed up once a year. We assess child cognition, psychopathology, academic outcomes, mother-child interaction, and brain imaging at multiple time points. To give an idea on what brain imaging look like at birth and 6 months of age, here we show the DTI and T2 atlases generated from our own data using probabilistic atlas estimation method developed by our group. The first 2 rows shows the color map and fractional anisotropy images derived from the DTI, and last row shows the structured T2 weighted images. In the first 6 months of life, the brain grows dramatically in terms of the overall size. The white matter tracks becomes more coherent. It is challenging to image the brain using conventional T1 and T2 weighted MR due to a similar T1 and T2 contrast in the brain, green matter, and white matter. The reversal of the white matter and green matter images contrast occurs around 6 to 9 months of age. At 4 years of age, the brain is adult-like in terms of overall size, the gyroscopic patterns of the cortex, and major white matter tracks. As previously mentioned, the primary objective of the GUSTO study is to investigate the impact of environmental and genetic factors on the developmental origins of mental health issues in children. Extensive research indicates that prenatal maternal depression significantly increases the risk of psychopathology in children during both adolescence and adulthood. Our inquiry revolves around whether the neonatal brain retains memory of fetal exposure to maternal depression, and consequently, whether the brain of neonates born to depressed mothers exhibit similarities to those of depressed adults, particularly in the corticolimbic circuitry. Upon analyzing the neonatal and infant brain imaging data, we discovered that higher level of prenatal maternal depressive symptoms correlated with decreased fractional anosopy in right amygdala of neonates, along with increased functional connectivity between the amygdala and the medial orbitofrontal cortex, as well as ventral medial prefrontal cortex. These findings provide valuable insights into a neuronal substrates that contributes to the susceptibility to depression, which appears to develop in response to the influence of maternal depression during intrauterine life. Furthermore, we have observed the elevated level of prenatal maternal depressive symptoms are associated with increased volume of right amygdala and decreased functional connectivity between the amygdala and orbital frontal cortex in 4-year-old girls, but not in boys. Interestingly, this pattern of correlation is opposite to what was observed during infancy. This discrepancy could potentially be attributed to a shift in the functional roles of amygdala from top-down to bottom-up processes. Notably, the influence of sex-specific factors only becomes apparent after birth in the gastro sample. Our next question revolves around the potential biological processes that contribute to the impact of prenatal maternal mental health on child brain development. To address this question, the gastro study collected the blood samples of neonates to extract genotypes and epigenetic markers. We initially dived into the analysis of epigenetic data, which memorize environmental influences at the cellular level. Through our examination of this data, we made an intriguing discovery. Epigenetic markers surrounding the glucocorticoid receptor NR3C1 exhibited the strongest association with prenatal maternal depressive symptoms and neonatal amygdala volume. Activation of NR3C1 in the brain region, such as amygdala, plays a pivotal role in regulating the function of HPA axis, which in turn governs circulating cortisol levels in humans. The NR3C1 genes is susceptible to epigenetic modification, particularly DNA methylation of this promoter, in response to various environmental conditions, including prenatal maternal depression. Notably, NR3C1 promoter methylation serves as a predictor of gene expression and is linked to altered HPA response to stress, as well as effects on neural development. This indicates that receptor signaling plays a developmental role in regulating amygdala's maturation and response to stress. Next, we dive into the question of whether neonates with specific genotypes may exhibit heightened susceptibility to maternal depressive symptoms. Considering that mental illness is a complex trait influenced by multiple genes, we took a different approach in our study. Rather than focusing on individual genes, we select a set of genes associated with a certain level of risk for depression, as identified through previous genome-wise association studies. Subsequently, we utilized a mixed effect model developed in-house to investigate the interaction between the gene sites and prenatal maternal depressive symptoms on amygdala morphology. We observed that greater prenatal maternal depressive symptoms were linked to increased right amygdala volume in neonates with a higher genetic risk for depression. These results suggest a complex interplay between genetic predisposition and prenatal maternal mental health, influencing the structural development of amygdala in neonates. To further investigate the underlying mechanism, we focus on the genes that exhibited the most significant modulation effects. We then conduct a comprehensive gene network analysis and found out that the gene highlighted in yellow form a genetic network primarily associated with a glutamate receptor activation. And this network appears to play a crucial role in regulating the relationship between prenatal maternal depression and neonatal amygdala development. The identification of this key genetic network provides valuable insights into a potential molecular pathways involved in mediating the impact of maternal mental health on amygdala during prenatal period. And this finding can be replicated from a clinical sample acquired at University of California at Irvine. What about the late childhood and early adolescence? And this period is characterized by significant neurodevelopmental changes and offers valuable insights into an interplay between genetics and environments, and as well as their link with psychopathology. So we conduct analysis using the ABCD sample to investigate the pathways that connect environmental genetic factors with brain network and psychopathology in adolescence. Our examination involves analyzing structured brain network derived from diffusion-weighted images. The study includes a comprehensive set of 33 environmental factors, which varies from parental psychopathology, maternal substance use, developmental adversity, socioeconomic status, and aspects of school and family environments. In addition, we quantify genetic risk associated with 10 different psychiatric disorders, as well as total problems and cognitive ability. To assess child psychopathology, we utilize various measures, including CBCL, indicator of mania, and symptoms related to psychosis. We employ a structured equation modeling to examine the influence of these interconnected factors on brain networks and psychopathology in adolescence. Where 6 environmental factors derived from factor analysis were used. We found that 2 pathways link socioeconomic status, brain and psychopathology. The lower socioeconomic status was found to be associated with lower structural connectivity in the posterior default mode network and greater salience structure and connectivity, and with more severe psychosis and internalizing behavior in adolescence. Prematurity and birth weight were associated with early developed sensory motor and subcortical networks. But polygenic risk for psychiatric disorders had moderate effects on brain structure connectivity and psychopathology in adolescence. This finding suggests that a range of genetic and environmental factors can influence brain structure organization and psychopathology during adolescence. And addressing these risk factors may be important for promoting mental health outcomes in young people. In summary, the availability of large-scale population-based datasets has opened up a new possibility for discovering significant environmental factors and genetic markers that impact brain development and mental health. With this knowledge, we have begun to develop a supervised learning approach to identify individuals who may be at higher risk for psychiatric disorders. Moreover, we are actively conducting intervention studies aimed at maximizing the potential of children from early age. We invite you to visit our posters and join our oral sessions to learn more about our research findings and ongoing efforts in this field. Finally, I'd like to thank all my lab members who have contributed to the work I presented today and funding agency who has supporting on this research. Thank you very much.